virtual lab, I'm going to walk you through how to do DNA extractions. If you want to follow along, you're more than welcome to. We're going to be using common household chemicals like Dawn dish soap. Palmolive also works, but these two brands are the best. We're going to be using some chemical meat tenderizer, good old baking soda, and rubbing alcohol. And if you are following along, go ahead and stick your rubbing alcohol in the freezer. It's going to need to be cold for the last step. We're going to be extracting the DNA of wheat germ. Wheat germ is the embryo portion of the wheat kernel. The remainder is the endosperm, which is the food storage. So first we need to gather our supplies. We're going to use raw wheat germ. It's really important that this is raw. So during the heating process of toasting, we'll denature the DNA. And so if you use toasted wheat germ, this won't work. So going to need about a tablespoon of Dawn dish soap. Also, like I mentioned before, Palmolive also works, but make sure it's one of those two brands. You're going to need a little bit of ice to cool down your extraction toward the end. So make sure that you have a container that you can put some ice water into. We're going to make a baking powder solution. So we're going to dissolve 1 teaspoon of baking soda into about a 1/4 cup of water. You're also going to need a thermometer to make sure that your system is going to stay between 50 and 55 degrees Celsius, which is about 100 to 130 Fahrenheit. You're going to need a hot water bath, so you should have 1 or 2 inches of water in the bottom of a pan or whatever to heat up your extraction. But we don't want it to get too hot, so we're going to need to be able to monitor that temperature. You're also going to need a paper clip, and you're going to bend it into a hook so that we can pull out our DNA molecules after we've extracted them. The first stage of our DNA extraction is called the lysis period. So what we need to do is to break down that cell wall, the plasma membrane, and the nuclear envelope to get into where the DNA is stored at in our plant cells. Okay, so that cell lysis means breaking down the cells. We're going to add about 45 milliliters of water, which is about a 1/4 cup. Okay. We need to heat this up to somewhere between 50 and 55 degrees Celsius. That's 120 to 130 degrees Fahrenheit. Because I'm working in the lab, I'm going to use our hot water baths, but since you're at home, maybe you're going to have a kettle of warm water that you're going to heat up your water in. While I'm waiting for that, I'm going to measure out about a tablespoon of my wheat germ and about a tablespoon of my dish soap. So my water is at 50 degrees Celsius, so I'm ready for the next step. I'm going to take my about 1 tablespoon, or if you have a scale at home, 2 grams of my wheat germ, and I'm just going to sprinkle this right on the top. Now I'm going to measure out my tablespoon of dish soap and add that to my solution. Okay. I'm going to very carefully stir this. Stick it back in the hot water bath. It's going to be there for about 10 minutes. Like the phospholipids in the cell membrane and the nuclear envelope, the dish soap is amphiphytic, meaning that there are portions of the molecule that are hydrophilic and portions that are hydrophobic. The heat that we're applying is going to assist in the softening of that plasma membrane, and it's going to help this detergent work more efficiently. It's also going to denature any enzymes that are being released from that cell lysis that might damage the DNA. So during this cell lysis period, it's really important that we keep our temperature below 60 degrees Celsius. Higher temperatures are going to denature our DNA. It's going to split that from that double strand into 2 single strands. That will make it impossible for us to spool when we're at the last step and we're trying to observe that DNA. So we're going to keep our incubation period to a limit of 20 minutes, and we're going to make sure that our solution is cooled before we add our alcohol at the end. So just pulled my extraction out of the water bath. I'm going to use my 2 grams of meat tenderizer which is about a half a tablespoon. And I'm just going to add it to the solution. Also need to add 5 milliliters or 1 tablespoon of our baking soda solution. Okay. Mix it up, back in the hot water bath. Baking soda is sodium bicarbonate. This acts as a buffer to keep the pH of our solution near 7, which is neutral. DNA is the most stable at a pH of about 7. The reason that we add the meat tenderizer is it contains the enzyme peptin, which is naturally present in papaya, pineapple, and other fruits. The optimal temperature for the peptin enzyme is between 50 and 65 degrees Celsius, which is exactly where our extraction temperature is. And its optimal pH range is between 5 and 8, which is why we want to keep our solution about a pH 7. The peptin is going to complete the breakdown of the nuclear envelope, and it's going to remove some of the proteins that are currently coiling up the DNA, and then that's going to release the DNA free into our solution. The final step requires the addition of cold alcohol. DNA is hydrophilic due to the negatively charged phosphate groups. That means that DNA is going to interact with a partial positive portion of the polar water molecule, and it's going to form these weak hydrogen bonds. We add the alcohol, that's going to disrupt the hydrogen bonds between the water and the DNA. And the alcohol will interact with the DNA instead of the water. The DNA then will not be water soluble, and it will precipitate or become visible. And the alcohol will form this layer on the top of our extraction. I just pulled the extraction out of the ice bath. It's at 25 degrees Celsius or about 77 degrees Fahrenheit. And I'm now going to add 10 milliliters or 2 tablespoons of cold alcohol to my solution. Because I'm working in the lab, I'm going to use the stereological pipette. You guys are, 2 tablespoons is going to work nicely as well. Okay, I'm going to add this to the interface of my extraction. It's really important at this stage that we don't mix our solution, so we're going to keep it nice and stable and avoid any mixing. We want to create these different layers. So we have our alcohol layer, we're at the DNA at the interface of our water-soluble layer, and then we have our wheat germ solution down here. This is the DNA. We're going to use that paperclip hook to try to snag some of that DNA. Chapter 10 is going to be all about what do we do with this DNA after we're able to extract it. So if you followed along with this extraction, take a photo of your extraction, make sure it's in focus and it shows all the layers. Once you have your photo and your necessary emotional closure, you can just rinse this right down the drain.